All right, so hello everyone. It is my pleasure to welcome you all to the 7th Electrophysiology and Ion Channel Seminar. And we are very, very pleased that so many of you find these seminars interesting and you show up month after month. We really hope that we can maintain the really impressive attendance we've had so far. And we urge you, if you do like these seminars and if you find them useful, to spread the word to your colleagues and those that might not have heard of them. And so we really envisaged, I say we, I mean the SGP and the organizing committee, the series as part of, part sharing science and part community building. And by showing up here every month, all of you, both speakers and the audience, are contributing to that vision. So thank you for that. And speaking of community building, I don't know if you had a chance, but please check out the SGP JGP Junior Faculty Network, which is an initiative by the Society of General Physiologists and the Journal of General Physiology that connects young PIs with each other and more experienced scientists that can offer advice and wisdom in navigating the scary world of being a young junior investigator. So let's move on to today's seminar. So the last couple of EpIC talks focused on human physiology. And today we're taking a bit of a sharp turn away from that and into physiology of bacteria. So we're very excited to welcome Letícia Galera-Laporta, who is a postdoctoral fellow in the lab of Gürol Suel at UCSD. Leticia's work focuses on bacteria and asks some really interesting questions about bacterial proliferation and their behavior in polybacterial cultures and in biofilms. So her latest work, which I think she might tell us about today, looks at dormant bacterial spores and the key to bringing them back to life. So without further ado, I'll let Letícia have the word.
Okay. Well, first of all, let me thank the organizers for inviting me to this talk. I'm very excited about it. I think it's the first time I don't need to give an introduction about how important are ions in cells, which make me very happy. So let me start sharing my screen. Okay. Okay. Good. So I'm going to give like a very brief introduction about action potentials. So action potentials are these sudden changes of the ionic content inside cells that happens because there is this efflux of ions through ion channels. And the most classical example of an action potential would be in neurons, what happens in neurons. So we have that there is this opening of different types of ion channels, and then the neurons change their membrane potential and propagate the information through other types of neurons. And one of the most important ion channels that are used for to produce the action potentials are potassium ion channel, which was published the structure in 1988 by Doyle, who was in the group of Roderick McKinnon, who won the Nobel Prize because of the structure of the potassium ion channel. And this was almost like in a record time because the publication was in 98 and the Nobel Prize was like 5 years later after the publication. And I don't know if you know this piece of information, but I think it's interesting that when Doyle was doing the research, he found that the potassium ion channels was not extracted from neurons, not muscle cells. It was extracted from bacterial cells. And why is that? So the first reason is because bacteria are very easy to grow. So you can have like a high quantity of the protein. And most importantly is because ion channels, the structure of the ion channel is very similar from different types of life. Doesn't matter if it's from an eukaryotic cell or a prokaryotic cell. the general structure of the ion channel is exactly the same. And one of the things that caught my attention is that the structure of the ion channel was discovered in 98, but there was almost no information on what these ion channels do in the bacterial cells. So there is also this paper from Chin Kung. He is a professor in Wisconsin and in Washington. And they published this paper where they say in the abstract here that it is ironic that the puzzling basis of ion specificity is finally solved in concrete terms with a channel from a lowly bacterium. And it's funny that they like do this like lowly bacterium. And its true function in the life of the bacterium is unknown. So when I started my PhD and I was reading any type of paper and I was like seeing this type of word like unknown, like this was music for my ears, because I don't know, I felt that everything was discovered already, but no, there are many things still to understand. Now as a postdoc, I feel that almost we don't understand anything, but I guess that this is like normal phases of a scientist. So in the laboratory of Guroswell, we have been studying what are the possible roles that ion channels have in bacteria, and we have been having some like discoveries during the recent years. And for this talk, I would like to explain you the main project that I've been working as a postdoc in his lab. So let me start from the beginning. In the lab, we work with Bacillus subtilis, that it's a bacterial cell that, as many other microorganisms, when is under stress, they form these spores, that it's a specialized cell type. And I would like to highlight two of the main characteristics of this cell type. The first one is that they are dormant. And when I mean that they are dormant, is that they don't have any metabolic activity. They don't consume ATP, they don't generate ATP, they don't have gene expression. And some people consider them that they are like almost like rocks. And the second characteristic that I would like to highlight is that they are very resistant. So for example, there is this group that found spores in amber, and they were there for millions of years. And this group was able to revive these spores in the correct conditions. And also there is this other group that sent spores in the outer space, and they saw that bacterial spores can survive UV radiation. And most importantly, they are a major source of antibiotic resistance, as well as a major source of food contamination. So as you can imagine, there are plenty of groups that are studying spores, but especially they focus their attention in the sporulation process, that this is how the spore is formed when the bacteria is under stress. And then there are also plenty of groups studying the germination process, that is when the spore encounters the correct nutrients, that we call them germinants, they resume growth again, and they move to a dormant spore, to an active cell that can grow and replicate. But there is almost no information about what happens when spores are in this moment, when they are dormant cells. And I think that in here is where resides one of the most outstanding questions, that is like how spores decide when they need to like resume growth, how spores monitor their environment and make this important decision that is going back to life again. So to answer this question, we use microfluidic devices where they have this type of trap where we can put the spores, and then media can flow in both sides of the chamber. And we also use microscopy, and in here I'm going to show you a movie of phase contrast, where we can, because we work with these microfluidic systems, we can switch on and off different types of conditions. And in this one, I'm going to start the movie at time 0 when I switch on and introduce the germinant, that is what makes the spores germinate. And it will be in constant flow of this germinant. So we start the movie where we see a white spore because spores are dehydrated. And then because of refractability, we see them white. But then you will see that in less than 12 minutes, the spore moves from being white to being black. And this means that they have completely germinated. But spores, as many as all living cells, they are not in constant conditions. Bacillus subtilis is found normally in the soil. So as you can imagine, like the environment continuously change. So we wanted to know what would happen when we, instead of flowing continuous germinant, how the spore will behave if we have these germinant pulses that we did like every 3 minutes. We were adding L-alanine, that it's like one of the most used germinants for spores. And then we were spacing our pulses every 2 hours. Now I'm going to explain why. And we want to know what happens with this spore. So to answer that, we do different types of quantification and how we use the method that we use to determine if they are dormant or germinant is through the phase contrast intensity. So here in this moment is when the cell is dormant, and then they change the phase contrast when they are completely germinated. So we have like half of the phase contrast intensity is when we determine that they germinated. So now we can do a germinant pulse of 3 minutes, and we see that, and 3 minutes is just because of technical limitation. So we want to be sure that there is enough germinant in the whole chamber. And we see that in less than 1 hour, all the spores that germinated already changed their phase intensity. And we wanted to be sure that all the spores that were germinating in the first pulse already germinated. So we went to double time, so 2 hours, to be sure that when we do a second pulse, the spores that we see that germinate, it will be because of the second pulse and not because of the first one. And most importantly, we also can see that when we do a germinant pulse, the spores that germinated keep germinated, and then the spores that didn't germinate keep dormant for the rest of the movie. And here I was using 20 hours. So now I'm going to show you an example of a movie of spores germinating during different germinant pulses. And for that, I'm going to also show a plot where each line will correspond to one of the spores, and then we will follow them over time. And there will be some vertical dash orange lines. That is when the germinant is introduced. And then the line will turn black when the spores germinate. But I think it's easier when we see it. So these are the germinant pulses, and we see that they turn black when the spores are germinated. So having this type of data, we can analyze it. And in here, I'm showing you, this is the dormant fraction versus time. So at the beginning of the movie, we have that 100% of the spores are dormant. And then after the first germinant pulse, there is a small fraction of spores that germinated. And then there is the second pulse, and then another fraction of spores germinated. And we can do this type of analysis for thousands of spores. From this type of plot, we can also show the data in a different way. And in here, I'm going to show you the germination probability. And this is because the first question that we had is that, do spores integrate information, or are spores just like sporadically or stochastically germinating when they encounter this germinant? So we would expect that if there is any type of, if there is no integration of the information, we should expect that for each germinant pulse, the same amount of spores should be germinating. But then what we obtained from this experiment was that we see an increase in the germination probability for the second pulse compared to the first one. So this type of change between the second and the first pulse is telling us that there is some integration of the information from the cell, and this is what we call the integration capacity. But again, like, how is it possible? How spores can be integrating information if they are this type of dormant cells? So we were wondering about this question for a long time. And one of the things that caught our attention was that spores are packed with ions. So because the cell is dormant and they don't produce ATP, they don't use the ATP, But the thing that they have free are many, many ions. So maybe they could use ions somehow to integrate this information. And then we thought about how neurons work, where there is a neurotransmitter that comes, binds to the ion channel, and then there is this sudden efflux of ions that leads to an increase of the membrane potential, a change in the membrane potential, and then it reaches a threshold, and then there is the spike of the action potential. So inspired by this example, we try to apply the mechanism that is called the intermittent fire response that is used widely used in neurobiology. And we apply this one in our spores. So we have in here a schematic where I'm showing a spore where we have the intracellular potassium and also the extracellular potassium, and then we have the conductance of this ion. Then changes in the ionic composition will have a change in the membrane potential. So in our model, we consider that the initial potassium content and the potassium channel conductance are the ones that are determining the potassium content dynamics, which at the end will tell us the germination propensity of the cell. Let me show you this in a different way. Here we have the germination propensity, so how easy for the spore is to germinate, and then we have it over time. So the spores reside in a certain germination propensity that is marked by the initial potassium content. If the spore has a lot of potassium, it will be farther low in the germination propensity. If the spores have less potassium, it will be closer to the germination threshold. And then how easily they approach to this germination threshold will be due to the potassium efflux. So we had this model and we did the simulations, and the model could reproduce well the experiments. But now that we have this type of model, we needed to validate it. And the main idea of the model is that spores are not like rocks. They can be acting as capacitors. So we have that the spore could be building all this energy in the form of potassium inside of the spore that then can be released in order to make work. So to test our model, we could test like the two main important pieces of the model. One is the initial potassium content and then the potassium efflux. So in here, I'm going to go over the different experiments that we did to corroborate our mathematical model. So the first one is we knock out the potassium importer that it's called KTRC. So what we expect by removing this potassium importer is that the cells will have less potassium from the beginning. So they should be closer to the germination threshold. So here I'm going to show you a movie where in the top we have wild-type spores, and then in the bottom we have the mutant that doesn't contain the KTRC. And we will see the exactly the same plot that I showed you before with different lines for the spores, and I'm going to show when the germinant is added. So this is how the stream behaves. We can again quantify and see that the model also can reproduce the experimental results, where we see that KTRC has higher probability right from the beginning in the first pulse and also in the second pulse than wild type. But importantly, the integration capacity, so the difference between the first and the second pulse, is exactly the same than wild type. And this is because we are not changing the potassium efflux. We are only changing the initial potassium concentration. So how far or close are the spores in this from the germination threshold? Also, we did this experiment where we have the KTRC cells, and if we, during the sporulation process, if we add high potassium content in the media, then we would be loading the spores with more potassium. So we were expecting that by adding more potassium inside of the spore, we should be rescuing the phenotype. And what we could observe is that now that the spores contain more potassium in the interior, they are again farther from the threshold, and they behave in a similar manner than wild-type spores. So I have shown you how changing the initial potassium content can affect the germination propensity, but now I'm going to show you changes in potassium efflux and how this affects the behavior of the spore. So the first experiment we did was to block the potassium channel with a drug that is called quinine. And so from the beginning of the from the beginning of the experiments, we have spores that are exactly the same than wild-type. They are wild-type spores. The only thing is that during the experiment, we add this drug in the media. So now we are inhibiting the channel of potassium. So we are affecting this efflux, how easily they move towards the germination threshold. Again, I'm going to show you a movie, and you have wild-type plus quinine at the bottom. And already from the beginning, we can see that the presence of quinine is reducing the germination probability of the spores. So again, we can quantify this and also look at the model, and the data seems to reproduce well the behavior. And in this case, we have that the germination probability it's reduced in the presence of quinine, but more importantly, the integration capacity is changing this time. So we have that spores in the presence of quinine really have a lower integration capacity compared to wild-type spores. And again, this is because we are changing the efflux of the potassium ions. We also tested the HUGO potassium channel. This is one of the channels that are expressed in spores. And in this, for this mutant, we have a little bit of more complex behavior, because from the beginning, the spores have less potassium in the interior, so they are closer to the germination threshold. And also because the spores are missing this channel, now there is the efflux of potassium is also affected. So we have these two different things that are changing with this strain. So here we have the movie of HUGO and how it behaves. And again, we can quantify the results. And we can clearly see that HUGO at the beginning of the experiment is germinating much a higher fraction of the spores, but then is reduced for further pulses. We can quantify the germination probability, and again, we see this cross moment between the first and the second pulse compared to wild type, and the integration capacity of HUGO is affected. And again, this is because the first thing is that HUGO spores start with less potassium in the interior, so they are closer to the germination threshold, and then the efflux of potassium is affected because they are missing this important potassium channel. We also quantified the concentration of potassium inside of the spores, and we corroborated our idea that HUGO and KTRC have lower potassium in the interior from the beginning compared to wild type. This is the initial intracellular potassium. We were using this dye APG4, which stains the interior of the spore depending on the potassium. And also we could measure the signal and extrapolate the potassium concentration of these different strains. Finally, for this, to check the changes in potassium efflux, we use like a very simple experiment that is just quenching the potassium efflux by adding more potassium on the exterior. So if the spore encounters in a media where there is more potassium in the exterior than in the interior, if the channels open, there will be less efflux of potassium. So, and in here we could see that by increasing the concentration of potassium in the media, we were reducing the integration capacity because we were affecting the efflux of potassium. We also did one control where we were checking if it was something with osmotic pressure by using sorbitol, and we could see no effect with in this case. And comparing these changes in the flux with our strains, we could see that we could change the potassium efflux chemically or genetically. In both ways, we were affecting the potassium efflux and then the integration capacity of the spore. So far I have shown you that by changing the two main factors of our model, that is the initial potassium concentration and the potassium efflux, we are affecting the germination propensity of the spores, but more importantly, we are also changing the integration capacity of the spore. So in our model, we are saying that when the spore encounters these germinant pulses, they will be releasing these potassium ions into the medium. And this is what will make them to move towards a germination threshold. So by definition, if you are effluxing potassium ions to the exterior, you are making changes in the membrane potential. And this is what our model was capturing. So we moved to this, we use this THT dye that is a positively charged dye that has been reported to show changes in electrochemical potential of the spore. So what it does is that this THT binds to the coat of the spore and it reports how negative is the spore. So like if the spore becomes more negative, then we have more and more dye staining the coat of the spore. And in here, I'm going to show you a movie where we have, this is the fluorescence that is coming from the THT, this reporter of electrochemical potential. And in this small inset in here, I'm going to show you the face of the same spore. And I would like you to pay attention for both, because we will see changes in the electrochemical potential, but I want also to highlight that we won't see changes in the face. So spores are maintained dormant. And at the same time, I'm also going to show you this plot where I'm showing the electrochemical potential, the signal of the THT over time. So let me play this movie. So in here, I hope you could see the changes of the electrochemical potential for each germinant pulse. So for each of the dashed lines, we were introducing this germinant, and we could see that there were these jumps in the signal of THT. So there were jumps in the electrochemical potential of the spore. But most importantly, these changes didn't happen in phase because the spore remains dormant during all this process. Here I'm going to show you 3 different spores, also again with THT, but I would like you to pay attention to the neutral THT. So we are very lucky that in the same building we have this collaborator, Theodorakis group, and they synthesized for us this version of THT, which lacks the positive charge. So in this one, we are expecting that cells, this dye should not be reporting changes in membrane potential. And in this movie, we can see that even we are having these germinant pulses again, we cannot see the changes in electrochemical potential that we were seeing with the original THT, showing again that this dye is reporting the electrochemical potential of the spore. We also did another control where we were using TMRM, that is another known dye that reports electrochemical potential. And again, we could see the different changes in the signal when we were doing these germinant pulses. And another control that we wanted to do is to test if other types of germinant were also having this type of effect in spores. And in this case, we were using L-valin, that is another type of germinal. And by reporting also with THC the electrochemical potential of the spore, we could also see that for each germinal pulse, the spores were changing the electrochemical potential. But now we wanted to directly see if potassium was going out of the spore, because this was our main idea. So for that, we used APG4, but this time is not an APG4 that enters into the cell, it's an APG4 that stays outside of the spore. And in here, I'm going to show you a plot with the signal of APG4 that will report on the amount of extracellular potassium that it's surrounding the spore. And I'm going to show you this over time. And these come from single cell measurements. And again, we could see that for each germinal pulse, we were having these increases in the extracellular potassium, confirming that spores, every time that receive the germinal pulse, are effluxing potassium outside of the spore. And we also wanted to see in the addition of quinine, that is the drug that blocks potassium, we could see that there were no the changes in potassium didn't happen this time, saying that indeed it was potassium that it was going out of the spore when there is the germination. So to summarize a little bit all the data, here we have this plot where there is the integration capacity versus the average change in the electrochemical potential of the spore. And we can see that the strains and conditions that were having a bigger integration capacity are the conditions where we see a bigger change in the electrochemical potential. And this corroborates that then spores are using this potassium efflux to integrate the information of the environment. So this is our idea that spores are dormant and they encounter all these germinal pulses. And then what happens is that the potassium leaves the spore, and this makes changes in their electrochemical potential. And this is the way that they use to integrate all this information. And then this potassium efflux moves them towards a germination threshold. And when they reach this threshold, they fire the process of germination, and then spores go back to active life. And if you want to know all the details about this work, I would like you to take a look at this paper that we published in October last year to see further controls and more details explaining this story. So what are the implications that we have here? So first is that this is the first time, as far as we know, that someone has reported any physiological process in dormant spores. And what we have shown is that they use this type of integrate and fire mechanism that allow dormant cells to process information without the necessity of energy consumption. And maybe these spores could use this release of ions to kickstart some biological processes, but this is something that we still don't know. And as for food of thought, maybe these type of processes that are so general, that are using ions which are so prevalent in all different cell types, maybe other types of dormant cells could also use these electrochemical potential changes in order to process information. So to finish the talk, I would like to go back to this paper, and maybe now it's not completely unknown what ion channels do in microbes. We are now starting to understand what would be the function. So we just opened the box and we have started to understand a little bit more what is their function. So with that, I would like to thank all the authors from this paper, especially Kaito Kikuchi, who is also a first co-author on the paper, Colin Weatherwax, that was the one that developed the mathematical model, also Ashley and Professor Gurol Suel, and all our collaborators for helping to have this project. And with that, I would like also to thank you for listening to me. And now I will be happy to answer any question.
Right, so as usual, we'll take any questions in the chat, or if you would prefer to ask the question in person, please raise your hand, and then Greg can make you a panelist so you can ask your question directly. All right, so we have our first question here. So Rohit Kumar asks, I did not understand how your thioflavin-induced changes in electrochemical potential are specific related to potassium channel in question. Could you please elaborate and clarify if it was used to generate an initial readout or it was specifically pointed to potassium channel? That was the first part of the question.
I see. So let me repeat the question and see if I understood well. So the question is about if THT is reporting electrochemical potential or if it's reporting potassium changes, right? This is what I understood.
Yeah, yes.
Okay. So first, both things are linked together, right? Like if you change the potassium content, you will be changing the electrochemical potential of the cell. So potassium will be leaving the cell, and then the cell will become more and more negative. And then this dye specifically is known to report electrochemical potential. So it will bind on the spore as the spore becomes more negative. But then it's becoming more negative because of the potassium leaving the cell. But we can like directly measure the potassium going out of the cell. We can measure it with this other, let me see, with this other dye. This one is specific for potassium. So the APG4 reports directly on potassium. And we could see also that there were changes in the potassium content of the spore. I hope I answered this question.
Rohit, I hope that answers the first part of the question. And then the second question asks, the second part of the question asks, recently a significant change to plant growth are demonstrated by controlling the electric potential around the roots of the plant. Do you think there can be a role for spore germination there?
Yeah, I would say that, yeah, I mean, it's just a guess, but Bacillus subtilis is found in the soil and it lives close to roots. So, and also, so yeah, any change in the ionic content around the spore, I think that it will have an effect on the germination. And also it's interesting that as root plants release alanine, that is the main germinant for spores. So I'm sure that there is some connection between roots and how spores germinate. I'm sure of that. Yeah.
Right, so the next question from Augusto Ortega says, cool talk, can spores restore the intracellular potassium concentration if they don't germinate after one pulse? In other words, if you increase the time between pulses, do you see a different germination probability with each pulse? Interesting question.
Yes, I really like this question. This is a puzzle that still we have. So we can see that the spores do not, so they germinate differently if we separate the pulses for longer times. We can see that the spores somehow can recover. The mechanism, we don't know yet. So this is one of the things that are still unknown, which makes me like excited about the future. But we think, so from the preliminary results that we have, we think that it's not potassium going in, but we cannot tell you yet how the spores recovered from previous pulses. Something I didn't know.
All right, so in the next question comes from Artem Kondratsky. It says, fascinating story, really very interesting. Congratulations, Letícia. How about other ion conductances in the spores? Maybe something like sodium. Can that influence electrochemical potential?
We tested that. We tested like we did this experiment, several experiments where we were trying to quench different types of ions. So instead of like adding potassium in the exterior, we were like trying sodium, we tried magnesium, we tried calcium, we tried like different ions, and we didn't see this dramatic effect that we see with potassium. So we think that potassium is the main ion going out. We cannot discard yet that any other ion can be leaving the spore, but we know for sure that potassium is the main one.
Letícia, have you measured intracellular calcium levels? Because as you hyperpolarize, you would increase the driving force for calcium into the cell. I'm biased and always think calcium is mediating the downstream effects, right? That's the true signaling molecule. What do you think about that?
So spores are like a very special type of cell because like vegetative cells have a low concentration of calcium when they are like actively growing, but when the spore germinates, they increase the amount of calcium. Like I cannot tell you right now what's the specific concentration, but they increase a lot the calcium concentration. So I would say that it doesn't increase calcium just because the spore is loaded with calcium. So it would be against concentration. So for that, they would need ATP consumption, and there is no consumption of ATP.
So there isn't an inward driving force on calcium ions in the spore. There is not an inward driving force on the calcium ion because the intracellular calcium is so high. It's in millimolar, right? Mm-hmm. Wow. Okay. Yeah. Thanks.
Mm-hmm.
Next question comes from Damien Bell. It says, lovely studies, Letícia. Thank you. These spores fluxes are presumably via ion channels. The size of spores are too small for patch clamp, as is potentially the spore coat material not possible to seal. Is it possible to express the mammalian cells, or would the mammalian phospholipid membrane be so alien as to make it either very hard to express or not close to spore coat that the function would be altered too much?
Yeah, I hadn't thought of like this type of question. Yeah, I guess that it would be too different. But it would be interesting. Instead of like trying to reproduce what's happening in a spore in a dormant, like in a eukaryotic cell, maybe it would be interesting to find examples of dormant eukaryotic cells and see if they use the same type of mechanism to process information. That would be like, yeah, very nice to see. Like, and also like even in seed plants, like any type of like dormant cell that you could think of, it's something easy to test. And yeah, I think that this is something more general, not only for spores.
Next question from Irene Liu. Great talk and beautiful work. If I understand correctly, the efflux of potassium channel would make the membrane potential more negative. Mm-hmm. How would this mechanism contribute to the final action potential-like event to trigger to derive the spore? Thank you.
Yeah. So we have that there is this increase in the electrochemical potential in the spore for each germinant pulse, and then they reach some threshold that triggers the germination. We don't know exactly what would be the link between like reaching this threshold and activating the whole germination pathway. This is something that we don't know. This is an open question. Again, this is like just the beginning of something. Yeah, I'm glad that people has this type of questions. Yeah.
So, guess along the similar lines, Zhang Gao says, very nice talk. What could be the link between L-alanine treatment and potassium channel opening?
There is, so we have seen that the Hugo channels are the ones that are open when the cell is dormant. So we can see that every time that we introduce this germinant, there is this potassium efflux that it's through the Hugo channel. But also there is this recent work from Radner, where they corroborated our results, and we saw that during the germination process, this L-alanine activates the receptor of L-alanine, and this is a potassium channel itself. So yeah, there are like these two different processes. So one, I think that is when the cell is dormant, that would be using some type of ion channels. And then when there is the germination, it could be activated by the receptor of the L-alanine.
There's a question from Jose Carlos de Lima. Have you investigated any potassium influx implicated in germination?
No, we haven't yet. Like we have some guesses, like all these changes in the electrochemical potential. I'm sure that the spore uses it somehow. It cannot be like it's like for the production of ATP, right? Like the cell uses these chains of ions to generate energy. I'm sure that the spore is also using this efflux of potassium to do something, but we still don't know. We still don't know.
Here's a question from Bertil Hille. Bacteria probably use the electrochemical driving force on protons as the plasma membrane, at the plasma membrane to synthesize ATP.
Exactly that.
Is the hyperpolarization in the right direction to initiate ATP synthesis?
Yes, it is. Yes, it is. It is. This is something that I would really like to continue working on, like understanding if this type of change is, for example, generating ATP. Because for me, it's like the most like clear thing that it could be happening. Maybe I'm wrong, but yeah, it is a guess. Yes.
Another question. How did you do these very long experiments over 16 hours?
Oh, these ones are short. For us, these are very short experiments. So in the lab, we used to work like we work with biofilms, and these movies they take like 3 days, even more like 5 days. You can be growing a biofilm. So I was very happy when I moved to work with the spores, and they are like 24 hours, and you can have all your data. So for me, it's short. So I guess it depends on which type of cell or organism you are using. But yeah, we have the microfluidic device. So we have the cells in there. We have the microfluidic device has wells, and you put the media inside. So there is this media constantly flowing, and it can be days and days in a stable condition. We also have an incubator where we have the microfluidic chip inside. So the temperature is constant. You have constant flow of media. And that's the thing that we use. Yes. Yeah. Imagine like people that work with mice. For them, the experiments are even longer. So yeah, I'm not complaining that my experiments are 3 days long. Yeah.
Another question from Faye Clever. Are potassium channels, UGO, more highly expressed during dormancy versus other channels and versus after germination?
So the question is if UGO is more expressed during dormancy than in vegetative cells? Is this the...
If it's more highly expressed during dormancy than other channels and after germination?
I mean, as Bacillus subtilis have several ion channels, and not all of them are expressed during the process of sporulation and then the spore. We checked, like there is this paper where they did the proteomics of spores, and we know that UGO is expressed during a spore, but UGO is also present in a vegetative cell. Not all channels are expressed in spores, but UGO is expressed during vegetative and spore and in the spore form.
I actually had a question, and this may be something that I just missed in your talk, but how do you force these cells into dormancy to begin with?
Okay, this is indeed. I have a movie. If you want, I can show you, which I think it's I really like it. So let me show you this one. So let's see. Okay. So this is a movie where you can see different vegetative cells, and in here I'm highlighting just one cell. And when vegetative cells are stressed, like for these bacteria, for example, when they don't have nutrients, when they are in the presence of antibiotics, they can do these spores. And this is like the sporulation process. So as you can see, like the spore is formed inside of what we call the mother cell. And eventually the mother cell lyses, and then it releases this spore in the media. So this is like the general process of sporulation. I don't know if you had like a specific question of it, or.
Well, I was wondering if I guess someone did already ask this question. If during this time, you have, I guess you have an influx of potassium into the cell.
Yeah, I mean, potassium is it's the most abundant ion in vegetative cells, and the formation of the spore is inside of the mother cell. So potassium is also high in the spore. It's about the same concentration that in the vegetative cell. It's around 300 millimolar to 250 millimolar of potassium inside of the vegetative cell and the spore. So there is no like active influx. It would be more like the formation.
Concentrates. Okay, yeah. Okay, I understand.
Yeah, it's different for calcium. For calcium, because the vegetative cell doesn't have that much calcium, then there is an active influx into the spore to concentrate all the calcium inside. Yeah.
Okay, there is another question from Augusto Ortega. I'm wondering if the cells can teach each other whether they should germinate or not. If you artificially kill half of your population during germination, would this inhibit the other spores from germinating?
Because I think they should be affecting each other. Because again, like there is this flux of ions outside. And then if you have more ions outside, then it's affecting the flux of other ions from the other spores. And also spores are formed usually inside biofilms, where there is all this matrix that it's, so everything is contained. So I'm imagining like there are like a spore touching each other, and then there is all this matrix surrounding them, which is making like a barrier for ions to go out. So I'm assuming that, yeah, there must be some interaction between spores and affecting each other. Yeah, I think so. I don't know about the learning itself, but I think that there is some effect between them. Yeah. We don't know yet.
There's another question from Bertil Hillet. Asks, is there any electrophysiology on cloned and expressed UGO that tells us what regulates its gating?
No, it's not known. In the model, we assume that UGO is open through the presence of the germinant. This is the gating. But it's not known how the germinant can be affecting the UGO.
So I think still a lot of unknowns.
Yeah. A lot of future work. Yeah.
Thank you so much for a great talk.